Thank you, everybody, for joining us again today. I wanted to first remind you that when we started yesterday, I asked you all to think about committing to 3 principles: imagination, integration, and acceleration. And we've just heard a little bit about integration this morning. When I think about these 3 principles, I convert them immediately in my mind to 3 alternative concepts. Goals. What do we want to achieve? Process. How are we going to get there? And timely results. What will we accomplish and when? Because patients are waiting. Yesterday, we also heard about some of the promise that can be achieved when these are combined. So today, I'd like to share my vision for Cohen Veterans Bioscience and how we imagine the world that we seek to achieve. I would like to walk you through the challenges as I see them, ideas to tackle them, and finally, a roadmap ahead. When Steve Cohen and I embarked on this journey just last November, it's hard to imagine, my primary concern was, what could I possibly do with this tremendous generosity of funding that he's provided? What would have meaningful impact? Because after all, the DoD, the VA, the NIH had already invested better than $2.1 billion in these diseases. And what was an additional $50 million, as generous as that is, really going to accomplish? But I knew that though the brain is absolutely the most complex organ of study, we have all of the neuroscientists in our audience, and I know you know how hard it is. Often the scientific challenges we face are not the greatest obstacles to progress. And even when we have clearly defined goals, like the NRAP, and the greatest minds and expertise in hand and in this room, without organization and flexible capital, we can still fail. I emphasize that word flexibility because when I reached out to my colleagues, asking them, what should I do? I talked to government officials, I talked to my colleagues in academia and in industry, and what I kept hearing again and again, and actually I heard again yesterday as well after the end of the day, was that a mission-driven, patient-centric, nimble, and flexible organization could really add tremendous value to the field. So with that in mind, I started to develop our goals for the organization. So what are these? Admiral Losi reminded us about the continuum of care. And for us, and particularly for Anthony and I, this is represented by the translational map here. So T1 is the transfer of knowledge from basic science to clinical. T2 represents the transfer of these findings into practice settings. And T3 is the real-world implementation, the optimization of the clinical care model. So it's clear that Cohen Veterans Bioscience operates in T1. Anthony and Cohen Veterans Network operates in T3. And we have to work together on T2, the transfer between these two, in order to achieve our goal, which is personalized, precise, integrated solutions, the constellation of solutions, diagnostics, devices, all kinds of different interventions, including therapeutics. And I'm always struck that we talk about the continuum of care, but we never talk about a very fundamental engineering principle. I'm sure Doug will appreciate closing the loop so that we can learn from our experience and so that we can implement and iterate and do a better job as we go along. So in T1, the challenge is moving the basic science forward into clinical practice. And one of our major goals is to develop therapeutics. And I use that terminology of therapeutics extremely broadly, even though this is a slide that talks specifically about drug development. Really, it's the same map whether we're talking about drug development, device development, or any other type of evidence-based practice that we want to pursue. We have to go through a systematic process of review, and we have to go through a process of investment. Here, what we're looking at is the specific process of drug discovery as it's been executed by the pharmaceutical industry for a very long time. And it marches along from the discovery phase, where we take tens of thousands of molecules, we put them through preclinical testing, then hopefully translate that knowledge into clinical practice, what's applicable to a clinical human individual, and start testing it systematically for safety, of course, but more importantly, for effectiveness. And that happens in phase 2 and phase 3 trials. And what you're seeing is that this diminishing return over time. Why is that happening? This has been evaluated extensively by the industry over the last 10 or 15 years. We're failing because we're failing to predict the next stage. It's a failure of prediction. We are throwing tens of thousands of molecules at the wall to see if one will stick at the end. And we're investing billions of dollars to do it, just to get one. That is not the process that I want to follow. And I think, as you can see, there's no feedback loop in that process. Every single time we go from stage to stage, we're literally throwing the molecules over the wall to the next stage and the next group of individuals within our organization to take it forward. But we're not learning as we're proceeding and doing a better job. But our goal is to get to a therapeutic pipeline for PTSD and TBI. And as we heard yesterday, loud and clear from Dr. Crystal, there is no pipeline. I had done a landscape analysis to look at, I was hoping to show you a wonderful pipeline of molecules that are coming through. It's bleak. And I was talking to a number of our biotech partners in the audience last night, and they said, we need help. We need to de-risk this process. We need to improve the probability that we can get over to that goal line. So that's a goal. Our second goal is to be able to identify the individuals who should benefit and will benefit from our interventions. How do we know they have the disorder? How do we know which intervention we need to provide? How do we make it precise? The only way we can really do that is by developing an armamentarium of diagnostics. And so we would like to develop a portfolio of diagnostics that will address the different stages of disease. In this figure, what you see on the far left is the individual at baseline. Already, as Admiral Losi mentioned, has baseline risk factors, life factors, experiences, genetics, as Dr. Cohen showed us yesterday. All of these combine to give us a baseline risk even before we've ever experienced traumatic events. Then you have the incident trauma, and you might find yourself in an emergency department. How do they know whether you're going to be the person that walks out of there fine, 2-3 days later, you're back to normal life, you're doing well? Or you're going to be the person that 3 months down the line, as Dr. Marmor showed us, develops all of those behavioral readouts that we define as PTSD. We don't know. We'd love to have a screening tool that tells you who should we be monitoring and watching. We'd love to have an objective diagnostic test, as Dr. Marmor shared with us as well, to tell us that this individual has the disease, and furthermore, what type of disease is this? It's not one disease. It's complex. It's many PTSDs and many TBIs. It's that spectrum that Michael, I love that figure with the TBI spectrum. That's what we're dealing with across all of these conditions. And prediction of response to therapeutics and monitoring of the disease overall. Those are our goals. And that means we have to have a roadmap because it just doesn't happen automatically. Biomarker discoveries that our academic community makes every day, and I'll show you a slide shortly, thousands of discoveries are made, don't automatically get down the road to a commercial test. Part of those challenges are the ones that Michael shared with us and Andreas shared with us yesterday. Dr. Pena explained to us there's a complex regulatory process and it requires investment and requires us to replicate findings and then qualify markers and then develop the assays that measure those with high fidelity and so forth. Every single step has to be followed in a certain order in order to guarantee success. Every single step needs investment. We need to do this. And as I spent 15 years in the pharmaceutical research industry doing research, and as I look at the therapeutic pipeline, why are we failing there, I come back again and again to just 4 items. A lack of understanding about the fundamental causes of the biology of, that underlie these disorders, a lack of biomarkers and diagnostics to identify the individuals that we should be including in our studies, the difficulty of delivering drugs across the blood-brain barrier, not necessarily something that Cohen Veterans Bioscience is going to directly address, but the outcomes, the lack of quantitative endpoints to know that our interventions are effective is always with us. It's because of this list that so many R&D companies have stepped out of neuroscience. It's extremely risky. This is a high-risk business. We need to de-risk it. So that brings me to process. How are we going to do this? That's a pretty tall order of how do we address all of those challenges. And the first step in any process is to conduct a landscape. Understand what are we dealing with. So this is just a top-level biomarker landscape literature review conducted by one of my teammates, Allison Provost. We looked at a number of the major literature databases, the Integrity database that Thomson Reuters manages, the PubMed Central database, looking for all of the publications that talk about biomarkers in the psychiatric disorder space, and then breaking that down by where they claimed the biomarker was relevant for a given disease, whether it was PTSD, TBI, major depressive disorder, or suicide. And you could see the numbers. The hits are the number of markers that were identified. And you can see at the bottom how many of those made it down that pipeline from discovery to commercial regulatory test as of today. These are mostly molecular markers that we were looking for. So this does not talk about devices, some of the EEGs and brain scope type of things. These are molecular markers. But it gives you a sense of where we are starting from. Something's not happening. We have those discoveries. Why aren't they translating? The other part of this is, we talked about yesterday, translational models, animal models. The Institute of Medicine conducted a series of workshops of which I was actually a member and in fact helped to develop some of these workshops with Steve Hyman and others. And when we looked at translational models in that first workshop, it was pretty grim. We all sort of walked out of there thinking there's nothing here. Animal models, we have to just walk away. And we said we can't walk away from animal models. Animal models are fundamental. They're a way that we can look directly at the biology that we can't access in humans. So let's take a second look. And that's when we conducted the second the workshop to look at how do we do this better? How do we think about this better? And I think there are a number of ways that we can. The other issue that's plaguing us is the issue of reproducibility. And this is true in the biomarker space, it's true in the preclinical modeling space. And it was brought to light by Dr. Ioannidis back in 2005 with an article that he published which really lit up the field, I think. Why most published research findings are false. That's the kind of title that'll get you attention, and it did. And it caught the attention of the pharmaceutical industry because for many years we'd been working with academics and others and taking data in, findings in, and not being able to replicate them. And so finally, a number of our pharmaceutical company partners published those results, which also sparked incredible controversy until finally the NIH went out and did their own assessment and determined that, yes, we have a challenge here. NIH and Dr. Collins launched a reproducibility initiative now because we have to take a look at how we're doing the science. Dr. Kandel told us yesterday that one finding, is interesting. Finding it twice, that's a principle. That's reproducibility. That's robustness. That's what we need in order to move the field forward. And there have been wonderful efforts over the last several years. A number of organizations like the Society for Neuroscience, the NIH, Wellcome Trust, have gone in and looked at this issue head on and identified multiple factors that we can systematically address. So this is something we can manage, but we have to be aware of it because otherwise we will continue to do the things we did before, underpowering studies, not designing them with fit for purpose and so forth. The other challenge we're dealing with is the complexity of this disease, or diseases. And this figure shows you on the top level, we look at these disorders at the syndromic level. These are complex disorders that change over time. They wax, they wane. They change with environmental factors. Sometimes you're above this clinical diagnostic threshold. Sometimes you're sub-threshold. But at all levels, it's a complex map. And what we're trying to do is relate syndromic behavioral manifestations of disease now to a biological understanding. And that biology fits below the surface here, starting with the genetics and pathways that lead up from there, the interactome, which is just a catch-all word for genome, transcriptome, proteome, inflammasome, metabolome. Okay, what is that? That's the interactome when you put it all together and you map that out. Those are the pathways that are driving then the traits, the dimensions of the disease that then we define as the disease, the syndrome. So how do we get in there? How do we start to understand this biology at the fundamental level and connect back and forth between those different layers? How do we deconstruct PTSD? And we have launched an initiative working with many of you in the room, and we've had extremely dynamic workshop discussions, conference calls every 2 weeks as we start thinking about what are these constructs that underlie the symptoms of PTSD? What do we mean by intrusion? What do we mean by avoidance? What are these measures, things that we can measure in both the human and the animal, as Colonel Hack was saying yesterday? those things that can translate across are the things that are going to help us with our preclinical models. Those things that we can measure at the biological construct level are going to be robust biomarkers. We're not going to generate a diagnostic pan-PTSD biomarker. It's not going to happen. We're never going to get a panel of biomarkers that gives us one diagnosis. It's going to give us clusters of diagnoses. And what we want to know is what's your unique biosignature, and that's what we want to measure. So we have to together work through the relationship between these different constructs and then all of the different ways we can measure them and then translate them back into an animal model. And that's how we're thinking about it, and that's what we're trying to do in partnership with both the academic and the industry communities who are participating in this, as well as our partners from NIH who've also joined, and the DoD in these working group discussions. It's just been a tremendous experience because we have to think about systems modeling. The only way we can pull it all back together, we have to get away from reductionism, and we need to start thinking about the whole human. The cardiovascular component, I thought, there's another example. We think from here up in brain disease, and we forget that there are all of these manifestations from the neck down as well. We have to think about the microbiome and the way it interacts. We have to think about the environment. We have to think about ways that we can measure these factors, and this is where things like wearables come in. And therefore, there is a way to measure all of these domains. Today, if we bring technology forward and we collaborate in the right ways, we can build very high density data sets that include nanotechnology, wearables, measuring all of the different parameters that Dr. Bruner shared with you yesterday, as we simultaneously use the more traditional imaging, molecular omic measures, and then also utilize the mobile and social media tools. MindStrong tool, what a tremendous tool that is. Every day, and the things you do every day could diagnose you. And in combination, wouldn't this be the most powerful information data set that we could have for any disease? And if we had these data sets, wouldn't that change how we define them at a biological level? We can get these data sets. We're working closely with Dr. Marmor, NYU, Amit Etkin at Stanford to build a data set of 1,500 individuals with all of these blood and other biomarkers, deep phenotyping, deep imaging, deep molecular, neurocognitive, everything we could possibly measure in these individuals. And lots of blood, a lot of blood collection, so that we can look at the omics exhaustively. But we've learned from the reproducibility initiatives that power is everything, and especially when you're dealing with omic data, especially when you're dealing with big data. We're not going to get there alone. We're never going to get there alone. We need to start pulling together multiple data sets and scale the science. And we've been really fortunate to find partners who've led other very large studies who were interested to pool with us all of this phenotypic and imaging and other biomarker data and put it into a cloud-based, secure environment where we can share those data sets and take blood samples and run them on bioassay platforms that are valid platforms that are quantified, and then bring that all together so that we can deconstruct PTSD. This is the initiative that we're going to call Rapid DX, and you will hear more about that from others today. We're going to take all that data, and what are we going to do with all this big data? We've never dealt with data at this scale. We need to figure out a way of analyzing it, and that's using computational methods. And we don't know which computational methods are going to work because We've not done this before in this area. So we need to look at deep learning. We need to look at pathway analysis. We need to look at a variety of machine learning methods and see how we can understand how these different layers interact and how they predict the outcomes we're looking for. So model systems, the preclinical model systems need to be developed further. We need to validate them in terms of construct validity. There's an entire panel dedicated to this afternoon. I'm not going to say more about it, but they will. We need to think about computational approaches and how we can incorporate them into our process of understanding the biology. There's a whole session on that as well. So this is our vision. This is where we'd like to go. We started funding projects and initiatives. Some of these are legacy initiatives that we had already started through Stephen Cohen's foundation work. He had already funded NYU and Stanford to launch that large biomarkers initiative, and we adopted it. And then we launched with the Broad the genetics initiative. We've launched a tau in silico modeling program that you will hear about today. We've launched a CDISC PTSD standards initiative with the FDA. We've launched a wearables program you heard about yesterday, a brain bank, which you will hear about from Sabina today, the knowledge map, PTSD preclinical program, and the imaging library. This is just foundational stuff that we thought needed to get done, and we could see that because we had nimbleness and flexibility and we could bring different groups together in a way that only a nonprofit can do, we were able to pull these things together. And what's wonderful is the partnerships. It all comes down to people. How do we accelerate things? How do we get timely results? We need to have a mission-driven roadmap. We need to develop that map. I don't have that map today. I need to work with all of you, and I need to hear where do we need to go. There's already a map, the NRAP, but that doesn't go all the way through to the end goal we heard yesterday. There are things that we could do to add on to it, to build on it, to expand it. Let's do that. Let's develop some stage gates and milestones so that we ensure that we get there, not 10, 20 years from now, but tomorrow, as quickly as we can. You know, the science is slow. I know it's slow, but really a lot of times it's just us. Let's set some milestones and let's make sure we get there. And also make sure our targets are right. True goals, and I say true goals because as I shared yesterday, it's so easy for us to say, well, we can only get this far right now, and so we'll do incremental improvements, when we really need to say, this is really what we need to do, and this is how much it's going to take, and let's go find those resources to make it happen. And I'm always reminded by an anecdote I heard from Steve Jobs about hot cars. He was telling this story about what happens in engineering. You design this beautiful, fast, sleek car. It's really hot. And then you take that design, your roadmap, to the engineers, and they start, well, you know, we can't do this, we can't do that, and then they take it over to the marketing people, well, we can't sell this, and we can't sell that, and so on, and you wind up with a Ford Focus. That's not what I want. So, you know, it's a principle, right? We have to keep an eye on the ball and make sure our goals are right. So let's do that. And let's collaborate, because there's tremendous power in collaboration. I think you are all feeling it. I felt it yesterday. I felt it in every conversation I had, every great new idea that came up. If we can just find a way to harness all of that, that's powerful. And it's not easy to harness that. It's not easy to run enterprises like this. I've spent a lot of time trying to analyze consortia, alliance models of all kinds to see what works, what doesn't work. There are so many great examples out there. I tried over the years to pull together the best practices from many of them, and I cobbled together my own approach, and this is our cooperative alliance model. And I work with a very innovative legal mind, Ki Shin, who's in the audience today, who helps me craft, he says, the most unique legal agreements he's ever crafted. And he works with universities across the nation and industries across the nation. He says, this job is the one that makes him think, and he's always trying to figure out what we can do differently. So we've come up with all kinds of very unique and flexible arrangements, and case by case by case, so that we can have agreements that share the incentives across all of us. We have to change the incentive models so they're win-win-win-win. Finally, I have to thank the amazing team that I've just been so fortunate to bring to Cohen Veterans Bioscience. First, our executive leadership team. You've already met Terry. She's not only a member of our board, but she's an active member of our daily executive activities. She brings a lot of commercial marketing experience to everything we do. Allison Gage, who's on the far right, she's our scientific program director for a lot of clinical science and has 20 years of pharmaceutical industry experience. Andreas Jeroman, you've met yesterday with the diagnostics biomarker experience. And then Thomas Oberst, who's just a treasure. He came to me about 2 years ago. He had retired from Fidelity, been running their enterprise IT systems, and said, can I just help you guys? I want to just volunteer. And oh yeah, you can help us. And he's just done a tremendous job. Recently provided a report to me where he looked at 28 systems across the country for data sharing platforms using 40 parameters to figure out exactly what we need to move ahead. Tremendous expertise. And then our scientific program team, Patricia Kavitsky, Allison Provost, and Christoph Diaz. I know you know them. I know you talk to them every day. I know you're in those working group meetings, and I know you know what it takes to keep moving the ball forward is interactive program management. It's hands-on. That's why we can move things ahead quickly with just 3 people. They're constantly interacting with all of you, and you are the force behind all of this. I have an extended group of people we work with on a contract consulting basis, and they support our communications efforts, our legal and finance group, and I want to recognize them as well. So in conclusion, I don't want you ever to doubt, as Margaret Mead says, that a small group of very thoughtful, committed citizens can change the world. And indeed, it really has been the only thing that ever has. Thank you very much. So we actually have a few minutes for questions, if anyone would like to ask any.
Your systems approach gives a tremendous opportunity. Have you also thought about being mindful of the converse of PTSD, of using some of these resources to look into resilience factors?
Yeah, so we'd love to do that, and that's why I was really grateful that Admiral Losi reminded us again that we have to ensure that's part of the roadmap, and how we think about resilience and how we measure those factors. And I know Dr. Rothbaum has also raised this in the past, so absolutely something I think we need to all be thinking about.
Hi, Magali. Thanks
Hi,
for
Mark.
a great presentation, and it's an amazing vision and roadmap. It occurs to me that while listening that one of the big challenges that I'm not hearing addressed in much detail is is kind of the human resources and the skill sets that are needed to accomplish this kind of work. And in the field of PTSD, I know that there are precious few people who are capable of doing the kind of work that you've outlined, and even fewer trainees coming up through the educational pipelines that we have, which are traditionally clinical psychology and psychiatry and more clinical neuroscience. But even so, there's not a great pathway for developing the kinds of people who can really carry this work forward currently and going into the future. And I'm wondering whether Cohen can contribute to the training of the kind of people who would be able to accomplish this work, and what your thoughts are about pulling together the right kind of skills and dedicating them to PTSD and TBI.
Thank you for that question. It's a great question. I think that there are actually two components to that. First of all, this vision, this approach requires multidisciplinary participants. We need engineers and scientists and anthropologists. You need all kinds of people to come together to execute against something like this. And they're out there. And I'm always impressed by what happens when you bring different disciplines together in a room. And we've done this for some of the computational modeling work, and you bring a clinician together with a basic scientist and a software engineer and a bioinformaticist, and there's a spark, and magic happens. And it really is it's at that interface that it happens. So those individuals are out there. I think the individuals that we're lacking or that we need more of are the ones that are able to cross talk between the fields, and who can bring these people together and drive that dialogue. I think there is a reason why we have silos of disciplines, and it's hard to get across those. And there aren't that many people who have that type of training as you're suggesting. So it's a good opportunity to think about how we do that, and how we are, as an organization, also help build that. I do know that the Institute of Medicine recently did conduct a workshop on the next generation needs for advancing the pipeline of scientists and the field and developing people. And I think that's something that I'll definitely take back to Steve Hyman, who's still working on that report, as an interesting, it's a very important area that I don't think we even talked about there. Thank you. Martin.
A very down-to-earth, very specific question. The fact that there are people who look or who are resistant to stress in combat or so, that could constitute an extreme phenotype. Do you include that in the genetic studies? Because that could be very, very interesting to find what the genetics profile of somebody is who runs through hell and still survives.
Well, I think Harrison will be in a better position to answer part of that question, but the way that study was done, we are looking at both individuals who are cases and trauma-exposed controls who are not, at that point in time, cases. So in theory, it's possible to look at an extreme phenotype of having been exposed and having absolutely no symptomatology, maybe after repeated exposures or something like that. We could come up with a definition of what we mean by resilience. I'm sure Barbara would love to help us do that. And then look to see whether there is a subtype of genetic resistance. Yeah, there. I don't know if Kirsten is in the room, and Charlie would like to add comments. Yeah. Oh, Kirsten, go ahead. Oh, right behind.
couple people brought this up, a similar idea to me, and we can definitely do something like this with the data we have, and as we gather more phenotype data, because unlike some maybe other parts of the PGC, much of our data comes from more epidemiologic or cohort type studies. So they actually, I think almost none of our samples are sort of like small like PTSD cases and controls, are more from broader populations. So we do have quite a range of trauma and a range of symptoms. So I think that's exactly, and it would take some thinking to define. So if people are interested in thinking about it, we could define that, I think.
It looks like maybe you've got a number of folks there, Paula, and others who want to comment.
I just want to ask a question while Karsten can take the mic. This is incredible. Well, I'm going to ask you too, Magali. I love what you've articulated as the symptoms and the constructs, except where are psychomarkers in all of this? Because I would think those are relevant too, like experiential avoidance. And I wonder if the two of you could comment. It seems to me that that's going to reflect a piece that will help us with the understanding of what is the mediator between the underlying biology and the symptom that's expressed.
So I have a couple of thoughts on that. So one is where folks have collected that, we are gathering that now. We're requesting all the phenotype data, which is actually, that in itself is a huge effort. Some of these are, a couple of our cohorts have 5 ways of longitudinal data and things like that. So, and then the other thing I didn't mention yesterday at all is there's several groups within the PGC PTSD group. There is, this isn't exactly what you said, but there's a psychophysiology group, an imaging group, other biomarkers, epigenetics, and the goal is to integrate all of it and to bring it all together. We haven't done that yet, but that's where I totally agree. That's where we need to go.
So theoretically, if we can, if people will share all of those different data types and variables, then we would be in a position to look at that. And that's what I have to say, that the PGC as a consortia effort is just a tremendous resource, and it's been a wonderful group to work with. Everybody's so collaborative and so roll up your sleeves and let's get it done. So I think if anywhere, that's the place we can do it. And yeah, go ahead, Israel.
Just a comment to the question about resilience. So there is actually a relatively it's complicated technically, but conceptually it's not complicated. So the moment we do gene bioenvironment interaction models, which increase with that, and we show difference in trajectory, we actually can take a candidates of resilience. This is done in a smaller cohort. We published something in JAMA with a response to adrenal receptor 2B, which actually predicts a more resilient trajectory. So I think when the gene bioenvironment, G by E models can be applied in the large scale to PGC data, and we are going to do it at some point, then it will automatically address some of the questions of resilience, even without going on. And of course, as Karsten said, we have a lot of phenotype data that can be used as an additional resilience markers.
Okay. I'm being told that we need to move ahead, so we'll take one more question.
Thank you for this wonderful roadmap that you've shown, but for those of us who are interested in the timely results and engaged in on-the-ground therapeutics, one of the things that I was worrying about is that for those of us engaged in on-the-ground therapeutics, one of the historic deficiencies has been the artificial silos between addiction science and mental health, and the ramifications for that on the ground have been pretty disastrous in terms of getting people into recovery. So I'm wondering, in listening to all of this, what we might do to lessen the possibility of on-the-ground siloing for the care that we offer, particularly between TBI and PTSD. You know, if those are conceived of separately on the scientific view too much, we have to think of how that will play out when we're trying to run clinics and provide care. So I'm wondering what kind of safeguards to have some kind of holistic view that might be implementable for those of us providing care?
And I'd like to jump on that because looking at the user end, and there's a case that's going on right now actually with our Special Forces community with SOCOM on something that I was very supportive of and trying to get it out there quickly, trying to get it into the VA. And now that it's been out there, the protocols for safety were not there. And so now I have totally backtracked. So I worry when I hear, let's speed, let's accelerate. And substance abuse is one of those things that can have a drastic effect when you implement some type of therapeutic thing if you don't take that into
Understand
account.
the liability.
Yeah, so that's, I'd be curious on the acceleration and then looking at the holistic approach once it does get to the user.
Yes, yes, and to be clear, evidence-based acceleration, so we would know and understand those types of factors. So to answer those questions, first of all, just to be clear, we do not yet have a roadmap, but we have our initial projects that we've launched where we saw opportunities right away, and we would like to work with the entire community here to build a comprehensive roadmap and to expand on the existing roadmap that exists. We have been already approached by the NIAAA, for example, National Institute of Alcohol and Abuse, and Dr. Grandison will be speaking later today on this as well, that they also would like to see how can we bring these pieces together and look at comorbidity across substance abuse and drug abuse as well as PTSD and TBI. We've had a similar conversation with the NINDS about the comorbidity between PTSD and TBI. So these are things that we absolutely have to think about, and Charlie and I have been thinking a lot about within the cohort at NYU. How can we do a better job of expanding the recruitment, because it's slightly biased towards certain types of individuals, and we want to have a much more representative sample so that we have biomarkers that are representative of the entire spectrum. So I think this type of spectrum thinking, it's hard to do. You have to really think differently because we've always used classic criteria, and that's how we diagnose, and then we limit ourselves. So we actually really have to unpack this a little bit before we continue forward. So I'm very glad you brought that forward.
Yeah, and sometimes you have something that's already been approved by the FDA for use in a slightly similar area.
Yeah, and that's going to be some of the work we have to do together as well. So thank you for your questions, and thank you for your collaboration. Many of you already work with us every day, and I really appreciate all the time that you do invest. I'm always amazed. We have calls with 40, 50 people at one time. Somehow we manage to do that as a conference call, and they're very effective.